Good morning, class 12. So here we are on our translation. Next step. Last time we finished with that RNA was attached to the smaller subunit. Let's quickly revise our previous little steps quickly. That what was happening, that amino acids, they were getting activated, and then these amino acids, they were transferred to the tRNA, and they form a complex which is known as now amino acyl tRNA complex with the AMP and the enzyme. Now then we discussed about that how process of protein synthesis start with the assembly of ribosomal subunit. There are two subunits of ribosome, smaller and the larger. On larger subunit there is three sites, EPA. A is the amino acid receiving site, P is where peptide bond formation occurs, and E is exit site from where tRNA will move out, and smaller unit where mRNA is going to attach. That we discussed in our last lecture. And these two subunits only assemble in the presence of magnesium ions, as well as only when protein synthesis has to be there in the cell. So here is the last step we talked about, that was attachment of mRNA. Now as you can see that mRNA is on our smaller subunit. mRNA is attached, and this is from 5' to 3' direction it happens. So now when the polypeptide synthesis, now the next step is that polypeptides are going to be formed. Polypeptide synthesis that occurs. So how does this polypeptide synthesis mean? That amino acids, now one to many amino acids, they are going to join with the help of peptide bond. So in this way polypeptide, or you can say a chain with the many peptide bond, so polypeptide will be synthesized. There are few points, especially 3 points I need to mention here before I start. That each tRNA, we said that tRNA it will come here at the site, A site, and that receive the amino acid. This A site receive the amino acid. So each tRNA is specific for a particular amino acid. Means every, either is suppose methionine, it will have its own it will have its own tRNA. Valine, it will have its own tRNA. So every sequence of amino acid, every code of amino acid has its own tRNA, except who will not have the tRNA, except the stop codons. Which are your stop codons? That is UAA, UAG, UGA. They do not have their tRNA. So a specific tRNA, then now this point is clear that each tRNA is specific for a particular amino acid. A specific tRNA is for initiation, for the starting. Like you know that methionine is the start codon. So that tRNA is called initiator tRNA, and there is no tRNA for stop codons. Stop codons like UAA, UAG, UGA, for them there is no tRNA. So this is the assembly. As you can see that this is smaller subunit, larger subunit, and mRNA is attached on the smaller subunit. Now on the larger we have APA, APE. Now one thing, when the tRNA, this is now initiated tRNA, this is the starting tRNA, that because starting codon is our AUG. Now here is one exception, because I told you that the amino acid, they will be received at the amino acyl site, but here one exception is that AUG, our first codon, right? AUG is our first code, and it always received at the P site. So so tRNA, suppose this is our tRNA, right? This is variable arm, and there is the your loop. So this tRNA is having your F-methionine, because AUG code for methionine. So here on mRNA AUG is present. So this will be always received at P site. It will all others code like now UAC, these all other code, all other amino acid, they will be read, they will be we will read them from site A, or we will receive them from site A. So here as you can see that this methionine, this is initiator tRNA, it will be received at P site. On the P site there is a code that is AUG. Then there are other amino acid UUU. For UUU there will be this amino acid. Then another amino acid is present outside on the mRNA, so that will further join together. So first we have to concentrate on this AUG and UUU. So now it is clear to you that once methionine has to come here, it will not be received at A end, it will be always received at P end, and then next amino acid will be received by the amino acyl site. So this is how this chain formation starts. Now the next is when now chain formation process is started now. So ribosome, see now just there it is not mRNA which moves, no, it is your ribosome, and here is your mRNA. Now this mRNA, this ribosome will move. Suppose there was the sequence which was present on P site, now it will reach at your E site, and their second amino acid will reach at the P site, and another amino acid will join at A site. So EPA with the movement of ribosome. So your mRNA does not move, it is your ribosome which is sliding. Ribosome slides over mRNA from codon to codon. Like it is like this. This is my ribosome, smaller subunit, and now here is the mRNA. So now with one code, another code, third code, so this ribosome is moving, sliding over mRNA in 5-to-3 direction. It's like your sliding window. Your base is fixed and the upper part is moving, right? So same is with the ribosome. So now AUG was present at the P site, as you can, let's go back. AUG was at the P site, and now it will come to the E site because of the movement of ribosome, and there is a new amino acid on the P site, that is your UUU, and one more amino acid come in, that is the at the A site. Now when this AUG reaches at the E site, tRNA will leave, and this methionine will form a peptide bond, because now UUU, that is your second amino acid, it will form a bond, peptide bond with the amino acid 2. Now once this CAU, it will reach to the peptide side, it will form the amino acid bond with second. So second and third, they will form bond. Once the third will reach at the P side. So this is how peptide bond formation occurs. So the moment they will move further, and new code at the site A will be completely added. मतलब ये move करता रहेगा, जो P पे था वो E पे आ जाएगा, जो A पे था वो P पे आ जाएगा, and P पे आने पे, because it is a peptide formation, so they have formed the peptide bond, and करते करते there is a long chain of peptide bond occurs. So the moment any tRNA reaches at E site, it is known as your exit site. So now tRNA will leave the exit site, but it will not carry the amino acid. Amino acid will detached, or tRNA will leave its amino acid, and this amino acid now can form the bond with the other amino acid. Clear? At this point, this process, this step is called translocation. Now ribosome will move further. tRNA carrying amino acid, now it was on P side. This is second amino acid and this was at P side. Now the moment it will ribosome slide further, it will reach at E side, detach amino acid and tRNA will move from the E side. Now as you can see that there is a E side पे क्या था आपका AUG and there was forming a peptide bond. Now with the movement of this ribosome, this UUU that will reaches at the E side, then one more new amino acid now which was on the A, it will come on the P and then one more will be added here. Now this we will talk about now. So first, so the movement of the ribosome, there is the this code is readed at the E and the P side, and P side they will form a bond. Now here first they form the bond with the methionine and amino acid 2, and now the amino acid 3 is on the P side, so amino acid 2 will form a bond with next amino acid. And when this process will stop, when the chain of formation of peptide bond, when does it stop? The movement, the stop codon, that is your UAG or UAA or UGA, anyone which comes at your mRNA, that is in the sequence, the moment it reaches on A site, now this UAG is not carry any kind of tRNA and it does not code for any amino acid, so no tRNA will be received here, so this translation process will be stopped. So at UAG, no tRNA, no amino acid, so because it is our stop codon, so it will terminate the translation process. So if UAG is not there till that time, peptide bond formation will keep on continuing. But once the moment the UAG will be there, then that this translation process will be stopped. So as you can see, the ribosome slide further, then chain of amino acids will be formed. F-methionine, 1, 2, 3, more and more amino acid, they keep on joining. So in this way, polypeptide chain is synthesized and it terminated till the terminated codon, which is your UAG, UGA, UAA, right? UAA, UAG, UGA is present on RNA. So this is called our elongation step. First was the activation, second is the elongation, and third is your termination, right? So here the second was the elongation and this is the termination step. Elongation means your chain of formation of amino acid with the peptide bond, that is called your elongation. And last is your termination. This is called terminated with the presence of UAG, UGA and UAA. So there are few points in the elongation that a peptide bond is formed. Definitely a peptide bond is formed between the carboxyl group of amino acid at P site and amino group of amino acid at the A site. So when there is the moment the new amino acid will enter at the A and the first amino acid is suppose at the P site, there is the bond formation starts. So this bond is formed, this peptide bond CONH, which is formed between the carboxyl group and amino group. This is formed with the help of enzyme which is called your peptidyl transferase. The bond formed between two amino acid is with the enzyme, and that enzyme is called peptidyl transferase. And according to the sequence of codons like AUG, UUU, UAC, CAU, amino acids attached to one another by the peptide bond and a polypeptide chain is formed. Polypeptide means many peptide bonds, formation of many peptide bonds. So in the last there is a termination, and when the A site of ribosome, now there are the three sites EPA in the larger subunit. So when the A site of ribosome, this reaches at the termination codon, now on the A site there is suppose UAG. Now it does not code for any amino acid, no charge TNA. So now no amino acyl tRNA. So now there will be the termination of polypeptide chain. So release or dissociation of polypeptide chain takes place. Now see, there is a long chain is developing here. Amino acid 1, suppose I write first F-methionine, then amino acid 2, then amino acid 3, amino acid 4. In this way a long polypeptide chain is formed now, but still it is attached to the ribosome. So how does it detach from ribosome? It is catalyzed by release factor. A release factor help in detachment of this chain from the ribosomal subunit. So now in the translation, if sometimes even in the vivas, sometimes the examiner asks that what is so special about a translation unit, what do you need? You need specifically mRNA. It should be from 5' to 3'. Now this in mRNA there should be, now on mRNA we need one start codon. Definitely there should be one start codon. There is a area from which amino acids they are joined and polypeptide is formed. At the end it should have a stop codon, and then there is a new thing which we have added here, that is untranslated regions. See, suppose this is mRNA 5' to 3', and here is my start codon, here is the stop codon. So here stop and start codon, and this is the area from where polypeptide chain will be synthesized. Now before starting codon and even in the ending of starting codon, at the end of stop codon, before starting codon, at the end of the stop codon, there are present few sequences or few areas which are called untranslated regions. Untranslated regions, what are these? These are the additional sequences. So here these sequences are present, additional sequences of mRNA which are not going to translated. Now the question comes in mind that then why they are present? They are present at both 5' as well as 3' 5' means before start codon, 3' means after stop codon. Why they are present? For efficient translation process, because translation only start at initiator site and it stop at the stop codon. Before starting and after stop there should not be any translation of mRNA. So for that purpose, for efficiency, for maintaining the efficiency of translation process, what is present? They are present UTRs. They do not translate into any kind of protein. They do not participate in the translation process, right? So that is all about our today's we are completed with our translation thoroughly. Now in my next lecture I'm going to talk about the regulation of gene expression.